Hello, everyone. Thanks for joining us today for this episode of QBI TV. I'm Robin Marks. I'm a Senior Public Information Representative here at UCSF. And QBI TV highlights research and practices at the Quantitative Biosciences Institute here at UCSF. Today, we'll be talking about some important work that a team of ours, an international team of ours, collaborators, have come together to make the most of an opportunity offered by the study SARS-CoV-2. And what they've discovered about SARS-CoV-2 and antibodies associated, human antibodies associated with it, will help us guide treatments and future vaccines to make them as effective as possible. So if you have any questions as the program is going on, please feel free to put them in the comments section. And we'll answer them. So to start off, I'd like to ask our panelists to introduce themselves and say a little bit about each of your roles in this work. So let's start with Dr. Chang.
Hi, my name is Yifan Chen. I'm a professor for biochemistry, biophysics department at the UCSF, most investigator of Howard Hughes Medical Institute. I'm a structural biologist, and my lab uses a structural biology tool, particularly single particle Cryo-EM, to look at the structures of protein and protein complexes.
And Dr. Craig, would you like to introduce yourself?
Sure. My name is Charles Craig. I'm a professor here at University of California, San Francisco, in the Department of Pharmaceutical Chemistry. I work on protein engineering and target validation in both infectious disease and cancer, and more recently in neurodegeneration. And I've collaborated with Dr. Chang, Yifan, for the past over 15 years, and with Cheng-Yi Wang, who's speaking soon, for even longer than that. And great to be here.
Thanks, Dr. Craig. And now Dr. Wong.
Hi, my name is Cheng-Yi Wang. I'm a senior principal investigator at the Singapore Immunology Network under Singapore's A-STAR. My research interest is I have a background in chemistry and antibody engineering, protein engineering. And the research focus in my lab in the last few years focused on engineering therapeutic antibodies against infectious disease and cancer. Thank you.
Great. Well, let's dig right into our discussion here. So your research looked at the role of antibodies in preventing a phenomenon called syncytia, I think that's how it's said, which is found in patients with severe COVID-19. So Dr. Wong, can you explain what syncytia is and the role it plays in COVID-19 and why you guys wanted to study it in relation to antibodies?
Sure. Syncytia is a biological phenomenon in which cells and cells fuse together to form giant cells with multiple nuclei in it. And coronavirus has been known to induce syncytia. And I don't think that there's any hard evidence of how exactly that happens. But the hypothesis is like this. So when a cell is infected by coronavirus, like SARS or COVID-19, and the spike protein is produced inside the cell, and the spike proteins are used to package more viral particles. But some spikes also somehow get transported to the membrane, cell membrane, and anchor on the outside of the cell. And when the cells with spike protein expressed on their surface sees the neighbor cells that has receptor ACE2 on it, so they see each other, they meet, and they interact. And then the consequence of this interaction is that the membranes were torn and then eventually cell fused. And this is how people think what happened. So the interesting thing about this is that, like Robin said, people have found that the correlation between syncytial formation and severe disease caused by COVID-19, but actually SARS as well. So we are wondering, if a neutralizing antibody can neutralize the virus by binding to the spike on the viral particle, can this antibody also bind to the spike on the infected cell, and then also prevent a cell-cell infusion to happen? And this is a study that we did back in last summer. And interestingly, what we found is that our lead antibody 586 can not only neutralize the antibody and prevent it from viral entry to the cell, it also blocks cell-cell fusion step. And so this is really, really exciting. But what surprises us is that another antibody, 3D11, does a very surprising thing. It neutralized viral infection, but it enhances cell-cell fusion process. And this is very, very strange, very interesting. And then we think that perhaps structure is a way to help us understand what exactly happened. And this is and actually this became the main focus of our study, led by Yufan's group.
And can you describe what we're looking at here, this figure?
Yes. So we're looking at a treatment of cells with different antibodies. So the way the experiment was done is that we take the cells that we know that they express ACE2, and then we transfect the spike genes into the cell. So the cell produces the spike, and they also have a receptor ACE2 there. And then if you look at the far left column, when there's no antibody, no trypsin, and you see a marginal expression, a syncytia formation, or almost no syncytia happening. But when you trigger, induce the spike protein conformation changes by adding trypsin. Of course, trypsin is not a natural protease for starting this process, but experimentally we use a protease to initiate this process. And then you can see on the second column, you can see some green image with multiple nuclei in it, a few of them, not very many. But if we do this experiment in a condition where we add our lead antibody 586 IgG, and then it actually can reduce, it can prevent or maintain the syncytia at the basal level. Okay. And the next column, 586 Fab, which is only one arm of the IgG, doesn't prevent cell-cell fusion, but actually enhances. You can see that there are giant cells with 5, 6, or 10 nuclei in it, right? And the 3D11 also does the same thing. It enhances syncytial formation. Again, you can see a green image with a few blue spots there. So the blue spot represents the nuclei. And then and 2H4 in a way it's not as severe as severely inducing syncytia as 3D11, but it also does something like that. So the bottom line is that 5A6 antibody can prevent syncytial formation. 3D11 can kind of dramatically enhance the syncytial formation.
Okay, so this is very bizarre. How can it be that antibodies can both inhibit and promote this same process? Dr. Craig, do you want to speak to that one?
Sure, Robin. It's a really interesting question, and it's one that everyone always asks when they hear the story, as well as the reviewers who finally accepted the paper so it could be published in Cell. And to be honest, we don't exactly know. We're still actively involved in this research among the three of us, and we have some theories about it. So we're going to talk about the structure that Dr. Chang is going to talk about soon. But if you imagine the spike protein that has these receptor binding domains that are necessary to interact with the ACE2 receptor. So this is just the spike protein, and it has these receptor binding domains that can be in an up position or a down position. And our antibodies bind and can block those receptors from interacting with the ACE2 receptor. So that makes sense. You keep it from happening, you're going to block infection, and of course you'd block syncytia formation as well. However, as Ching-Yi was just talking about, 3D11 can now block infection, but it facilitates syncytia formation. And Ifan's structure, when he did the structure, he showed that 3D11 binds on the sides of these receptor binding domains, whereas 5A6 binds to the tips in both the up and the down position. Now that structural difference has to be part of the reason that when 3D11 binds, it can facilitate binding of this spike protein to the neighboring cell. And it can do it in such a way that it can now avoid blocking ACE2, but still bind to it. So that allows it to act as glue, whereas in the other one, it's acting to prevent it. Now it's even possible, and we have no data for this yet, but we're thinking, what if the spike proteins don't even need ACE2 to interact with the neighboring cell? That would allow it to cause fusion. And I was just reading a paper this morning about how these long-term COVID patients have kidney problems. And kidney problems, it's beyond the heart and the lung tissue that you see a lot of damage, but long-term COVID patients have kidney problems. And what if there's syncytia formation that's occurring in that case in the absence of ACE2? So these are all things that we're starting to explore. Long answer to your question, but it's a really exciting question.
Yeah, yeah, very interesting. So this all kind of comes down to the physical shape of the antibodies. And Dr. Chang, can you tell us how you and your collaborators were able to look at the shapes of the antibodies and determine the mechanisms that define their differing effects on infection?
Sure. So my lab is a structural biology lab. So we're basically trying to determine the structure of a complex protein. Complex is what's the shape. And as Charlie and Cheng-Yi mentioned that these antibodies, say, from chemical assay, you can characterize that they does, since they're both neutralizing viral infection, but then they must do something differently. For example, one syncytia, one inhibits syncytia. But what is the difference? Why they cause that? So that's why we want to do this, look at the structure. But then we can show this, say, of the structure as in the next figure. And you can see the structure was looks at the pretty complicated shape. Traditionally, determine protein structure like this will need to basically crystallize all these proteins into a crystal. And that basically means that a lot of these millions of these molecules, complex, then packed in a very ordered form, a very ordered crystal. Then you take this crystal to X-ray beamline to get the data and then to solve the structure. And this crystallization process takes a long time often. So that's why traditionally to getting the structure information of such complex will take a really long time. Now in this case, that we're using a technique called the single Cryo-EM. Basically, we bypass the requirement for crystallization. Instead, we freeze these molecules in this freestanding native conformation in this very thin layer of ice, and then putting this frozen sample into an electron microscope. We take a large number of images, and then we computationally join these images together and then identify identical molecules and join them together and then generating the structure. So one advantage is that this method is now very rapid, very fast. And as a matter of fact, that in this time, during this pandemic, that the first protein structure was determined only about like 4 or 5 years after the first viral genome was released. And in the comparison, in the previous one, that which was in the SARS, first SARS outbreak in 2003, that take years to getting those kind of information. So this information is shown here in this figure that really gave us more light to understand why these antibodies, besides neutralizing, but how why they look like differently. As you can see that in the middle row here, this is the 3D11, and you can see that all the antibody binding there and then binding the spike protein, in the open conformation. And also then the 5A6, the best antibody that both has high potency in neutralizing, but also inhibit the synthesia. You can see that lock certain this protein that in the down position, basically in the inactive or down position. And then we think that from this structure information, we think that this it is critical to maintain this protein in a down position to both preventing the viral infection for the neutralization, but also to preventing the synthesia formation. And we can talk more about why, for example, 3D11 can cause the synthesia for basically opens up this spike protein. And even when there's no presence of antibody, then you can see that it binds. This movie shows that generally, basically, this 5A6 shows that how does this 5A6 binds to the antibody, binds to the spike protein. You can look at this one as a really complicated shape. And one can imagine with a shape with so many things sticking out there, if you want to package this molecule into a crystal, it's incredibly difficult. That's in the past, the structural biology never able to crystallize such a large complex in this kind of shape with antibody bind. But using this new technology, single particle Cryo-EM, and then which we were able to really get into this level of the. And then the other thing we learned is that this antibody binding, not just simply think about blocking the viral infection by blocking the antigen binding to the basic ACE2 receptor, but it's also changed the 3D conformation. This is shows in the next figure, that which we can see that how the antibody binding, different antibody binding, that not only binds the protein, but it also change the conformation landscape. And it's showing here, for example, you can see that the 2H4 antibody, which Chang mentioned, that the bind changes the conformation of spike protein in a very similar way as the ACE2 and receptor binder. And then 3D11 basically accelerate this process. Also, it blocks the ACE2 binding, but this acceleration probably is the reason that make them more facilitating the synthesia formation. And in contrast, that 5A6 is that trap this protein in a very different conformation, which is incomparable to mediating the membrane fusion. So basically, this also led us to think about the antibody that not only just simply says considering how the antibody binding, but rather than also how antibody binding changed global conformation. And then so these are also the structural insight lead us to have a better understanding of how these different antibodies can function as both like a neutralizing antibody to prevent the viral infection, but also some of these antibodies, for example, 5A6, could be a good candidate to also for the treatment, even for the infected patients.
Thanks for all of that interesting information. I understand that Dr. Wang actually has a model of this. So I'm curious, how did the two of you work together on this model, or how did this model come about?
Right. So this is a model solved by Daniel in Ivan's group, and this represents spike trimer. I'm sorry, there's no color differences here. So the spike trimer here, and bound by two 5A6 FAB. So this is one antibody here, and a second antibody here, and this antibody binds to the up position of the spike, and this antibody binds to down position of the spike, leaving the third one unbound here.
Wow. That's pretty cool, I gotta say. So just wanted to remind you all that out there, if you have any questions for us, feel free to ask those questions in the comment box. And do we have a picture of Daniel, the model maker? He is looking proud of.
Yeah, Daniel is a graduate student and then he's the one of the main person that did the structure of this protein. But also another person to mention, the name is David Barkley, and he's the facility manager. And then this work was done during the pandemic because and also when the lab was all shut down. And David Barkley was the first person that we contacted, and then he operates all the instrument, and actually the first data set. And then even when the whole facility was shut down, and then he was there and then to operate this thing, and then he set up the whole pipeline so that it enable us to collect the data remotely, so that people don't have to come on site, but then they can remotely work the project. So those are the two people that they all are the co-author this paper. With Daniel is the first one, and also together with somebody from Chinese lab.
Okay. Do we, Chang, Dr. Wong, who is that from your lab?
Yeah, can you show the photo of Dr. Wambay, please? Yeah, so Dr. Wambay is the person shown to the right in this photo. And Dr. Wambay was born and raised in Wuhan. So when this outbreak started a year and a half ago, she volunteered to lead this project naturally, and she's been working on this project like 7 days a week for the entire year last year, or until like in the mid this year. And to the left is our research assistant, Chang.
I think we have a picture of them too.
So these are two people in my lab, Beatrice Wong and Nick Young, who, with the help of Marcus Bone, we can show a picture of separately. They were the ones who were making this spike protein. We'd initially started working with Ashish Manglik, who's another professor here, and he called around and he got the best construct for spike for us to start working with. And then we had to make reagent quantities of it. And this is them doing it in a tissue culture facility with their masks on, even though, you know, you couldn't do social distancing, but he was training, and then Beatrice is a senior graduate student who was training him how to do this. And it gives me a chance to just shout out how this collaboration worked pretty much 24 hours a day. I've frequently said that the sun never set on this meeting, on this project, because we were constantly working on it in one of our labs. Because Cheng Yi, being in Singapore, when he was getting up in the morning, we had already been working throughout his night during the day to get things to move along, and then we'd provide him information, and then his folks in his lab would pick up after that. And then Ifan's lab is one floor down from mine. So we were constantly doing things. Someone was always working on this 24/7. And then we'd have these pretty much daily updates through email and WhatsApp and various other social media to keep the information flowing. And then we'd have 1 week set aside, 1 day during the week set aside where we'd all get together on a platform where we could share data and do problem solving. And we were doing that starting, this project really started in January of 2020, but really kicked into high gear right during the pandemic when things were shut down. And we were all allowed to continue to work because it was considered essential work. So it's been one of those things that you got caught up in it and the sun never set on the project.
Well, and how did the three of you end up sort of coming together with various roles?
Yeah, this story goes like this. So when the outbreak started in December 2019, and there was evidence that this virus can transmit it from human to human, even as early as January last year. So Singapore suffered from SARS like 15, 17 years ago. So the government is very, very concerned about about this virus and this disease. So in January, A-Star management asked my lab to develop a neutralizing antibody against this virus, just in case. And so that was 26th of January, and the same day I sent an email to Charlie, and because I know that structure will be very, very helpful in terms of helping us understand or design a better neutralizing antibody against this virus. And so I asked Charlie whether he or his colleagues at UCSF can help us on the structure part, structural understanding of what we'll be discovering in the future. And Charlie sent an email back right away saying, definitely. And that's how the story started.
And you guys have worked together quite a bit, correctly, across borders?
Yeah, I was Charlie's postdoc back in 1992 to 1996. So I left his lab 25 years ago. And then I have this privilege that we have been working on and off together in the last 20-something years on so many different things. But COVID-19 project is the one that we worked together every day and very intensively and for such a long time. And it was a wonderful experience working with Charlie and Yifan in the last 18 months.
At what point did you bring Dr. Chang in?
Well, that's when I received that email and from Cheng Yi. And of course I went right downstairs and asked Yifan, because you see how important the structure is. And Yifan and I had been working for 10 years before that on leveraging the strengths of our two labs to do things that we couldn't do separately, to this synergy that 1 + 1 can become 3. And we've had some successes and some failures, and we're looking for another challenge. And so when this came up, I went down and I said, hey, Yifan, you heard about this virus? Would you be interested? And then I'll let him tell you what he said.
Yeah, so as Charlie mentioned, that our two labs have been working together for many years, ever since I started my lab at UCSF 2006. And we tried using antibody as a tool to facilitate structural determination. But this is actually the first time we're actually shifting the key to really focus on the antibody itself. January 2020, during the Wuhan shutdown, I was actually very concerned about it because I spent my 7 years in Wuhan. I was a undergraduate student at Wuhan University and also graduate student in Wuhan University for 7 years. And then I have many classmates, my teachers, and my friends, and then they live there. And so during the shutdown, we are very concerned about the situation there. We joined some efforts to make donations and collecting the material to ship there and then to support them. But also, so this outbreak to me is something more personal rather than reading something on the news. It's really that actually we have people we know are there, and then we saw the picture of somebody like a colleague in the published in New York Times in this big gigantic hospital lying on the bed. And so it makes us really connected to it and very concerned about it. So when Charlie came to me asking me, he said, have you heard? Of course, they're really concerned. So can you want to work on it? I immediately said yes. Because I always feel like there are two things. One is this personal connection. The second part is that we've been working on structural biology. And then we determine structure. I think that our structures are relevant to human health. And for the many years, we've been thinking like that. But this is really the first time I really feel like what our work is so close to the society, and then we feel like our efforts in the past many years, as a whole field, structural biology field, really made a big contribution to the society in fighting this pandemic. I feel very proud to be part of this work.
So yeah, both you and Dr. Wong kind of had a personal, Dr. Wong, was your lead scientist, was she in Wuhan at this time?
Yes, my lead scientist, Dr. Wambay, like I said, she was born and raised in Wuhan. So when she first heard about this outbreak and heard that A-STAR is asking us to develop neutralizing antibody against it, and we're all really excited. Of course, we feel sad about what happened, but so she immediately volunteered herself to lead this project, and at the end, she led a team of 13 or 14 scientists working with her, and so we were able to very rapidly identify antibodies against this virus in a matter of a few weeks. So we were preparing the sample, the antigen. We were producing the antigen, preparing all the reagents. So in early March, we started the antibody discovery work, and then within 2 weeks, we know we have something, right? There's evidence that we have something that binds to the spike and prevents the spike from binding to ACE2. So we were really excited, but not until that we produced the antibody and put it into the live virus neutralization experiments when we truly realized that we have something really, really promising. And that's the time when Charlie's and Yifan's lab is ready for the structural studies back in March last year, March, April. So the timing works out really, really well. So by the time we have the antibody and the spike production is ready for structural studies, that's really, really exciting.
So it sounds like there was a lot of power and strength in having this international collaboration. What were some of the advantages particular to this case, and what do you think are some of the advantages of working across borders rather than just within our borders in general?
I can make a start at that. I think having 3 different labs with 3 different strengths coming together is really important for any collaboration, right? You don't want to collaborate on something that that you're not bringing something to the party that's different than what other people bring, right? If everybody brings dessert, you don't have a dinner. You want to have multiple different parts of the dinner, and we were able to do that. The other is the aspect of the sense of urgency that was present here. It's true that perhaps one lab could do all of this, but why? Why do that? When there's people's, I mean, we're not trying to have delusions of grandeur here. I mean, we're trying to understand something truly fundamental. But of course, it's extremely exciting, as both Changyi and Yifan said, that if you can have something to do to help others with your research, it's an amazing feeling. because you're not trained that in graduate school. You know, we're not physicians, but working on something like this and wanting to do it very, very quickly is an enormous strength of collaboration, because then instead of you trying to figure out how to do it, someone else already knows how to do it. And working with both of these folks, I respect them both, but they are also very demanding. When after that first meeting I had with Chang-Yi, asking him and asking Yifan if he wanted to be part of this, well, our labs are one floor away, so when we see one another, he was constantly saying, well, have you got something? when are you going to get it? When are you going to get it? And so then Chang-Yi got it, and then we started making the spike protein, and so it wasn't like, well, we'll get to you next week. It was like, now, we want it now. And that just makes everything go so much faster. And of course, being able to work 24/7 also really helped a lot.
Yes, you can't ask your lab to work 24 hours, but now we have 2 different time zones, so they're all the 24 hours in a day, we have people working on it. So, yeah.
So because as Charlie mentioned, is this kind of urgency also put the community to, and then all the people in the lab, they're really putting a lot of efforts, and then beyond what, because people like in our lab, like Daniel and also David Barkley, and then they were at the beginning that what everybody's concerned with during the shutdown, that we don't really know how to operate this thing, and so Barkley was really came that he said, okay, I'm going to turn on the microscope and then maintain it running. I'm going to first just be in there myself collecting the data, but also set up all the remote control that enables not only our field, but many other studies carried out by the UCSF related to the QBI, some major consortium effort there, and then beyond that. And then also, I really think that this collaboration is much stronger than the individual one, and then in both the technique, in both the combined technique, different expertise, and also the morale that people really feel like we join together our efforts to fight for this disease, and everybody feel like they're making their own efforts to contribute to it. That part I feel like is also particularly strong in this. Yeah.
Great.
Yeah. That is definitely a, like, what did they say, that the sum is greater than the whole of its parts? No, the whole is greater than the sum of its parts. Yeah. Well, we have a question from the audience, and it is, I'm curious about the role of cell aggregation for the progression of the disease. Is there any causality there?
Charlie, you want to go?
Go ahead. You did the syncytial
okay.
studies.
So I like I said, I don't think there's a very, very hard evidence whether syncytia is the cause of the severe disease or is the outcome of the disease. But I think the evidence to show that it's the cell fusion process can lead to tissue damage. So there's evidence to show that the syncytia can be a cause for the severe disease. So curious about the role of cell aggregation for the progression. Yeah, I think more clinical data would tell us more about the correlation between these two. But I think the evidence is there. Any casualties? This clinical question, I'm not sure, but I'm sure that people who die of the severe disease, and I'm sure there are certain patients who do have the syncytia in their lungs. Charlie, I think you have some more information.
Jungmin, it's a great question, Jungmin, and it's something we think about a lot. And we referenced some papers in the paper we published where there's been absolutely a correlation. It's not causality. I know you're asking about causality,
Oh,
but
I'm sorry. Yeah.
the fact that there are, when you take patients that have died of COVID, and we didn't do this, but other groups had done this, and they look in the tissue, the heart and the lung tissue, and they stain it, they see these syncytia, these large multinucleated cells. And it's clear that there is damage there. Now, does the one cause the other? And that's the question that has to be addressed eventually to push forward. And we're trying to do it.
Min also had another question, which kind of leads into my next question too, and that is, his question is particularly, would it be important to use the right type of antibody, and can we control which antibody is generated in our bodies when we use a certain type of vaccine?
Yeah, so this is one we've been talking about, Jongmin, because so we're a little prepared for this one. And trying to keep it short, a couple points, and you really got the main point, and that is all antibodies are not good. And that's true in other infections as well. If you look at dengue virus infections, that second infection is really bad because some antibodies end up actually working against you in that case. So it's key that 5A6 is a good antibody. It's one that could be used right now for passive immunization in terms of these monoclonal antibodies that are used as treatments. 3D11, I wouldn't suggest is such a great one because of the possibility of it facilitating syncytia formation. But now in terms of vaccine design, how do you generate the antigen so that you can make the good antibodies versus the bad antibodies? And that's where we feel this work could have its greatest impact. In terms of subsequent vaccine design, how do you get the antibodies to bind to the RBDs in the way that 5A6 can? They can bind it to both the up and the down position. And Yifan and Jingyi and I have an idea that we're testing to see if that can actually work. So you have to wait until we get some data, because we don't want to send out the wrong message here that we got it figured out, because we don't. But we have some really good ideas, and I know other people are trying to do this as well for vaccine design to get the right antibodies made and not the wrong ones.
And I think also this brought the importance of knowing the structure. And once you have the structure, then you can think about originally how to try to generate a specific type of antibody or design the vaccine so that the antibody will be biased towards a certain conformation and another than the other. So that is structural information providing that that kind of information that which is could be necessary for it.
I can give you one example, like the spike protein. The three tips are the structure very dynamic. Like Charlie says, it can assume a up or down position, and the up position is a position that can interact with ACE2 of the host cell and trigger infection. So if we have a way to design a spike, right, so that all the spike are locked in a down position, right, and then if we can raise antibody to lock the conformation in such way that they would not expose it to the up position, and that would be, potentially it would be a wonderful vaccine. And I think the structure that can, the tool that we can study the structure to see how we can design such spike will be very, very important. So that Yifan's technology will be very, very helpful for the vaccine design.
And our audience member also had another kind of addendum to that question is it valid to speculate that some antibodies can make the symptoms even worse and that we need to be careful about which antibodies we're generating?
Well, that's getting to the question of causality. I mean, we wouldn't want that to happen, But I think that's one of the questions that's raised by not just our studies, but other studies, that some individuals respond very well when their B cells are working well and they make good antibody responses and they do quite well. Other people don't. And is there a way to be able to tease out the good antibodies versus the bad antibodies? And that's going to involve really studying some individuals that have responded well and what were their good antibodies, and the ones who didn't respond well, what were their antibodies? So that would be one of the ways to get at that question.
And we've talked a little bit, or you guys have talked a little bit about the implications of this study for developing vaccines. What might be the implications of this work for treatments as well?
Go ahead, Ching-Yi. I think that's in your space there.
As I mean, antibodies as a treatment, right? That's your question, right? So when we started this discovery work, we did not think about the mutants or the variants will come so rapidly and become such a huge concern, right? So a therapeutic antibody design in the future, I think we should think about how we can look at conserve sites so that the antibody will have a broader spectrum of neutralization against possible mutations. And this is easier said than done. It's actually it's quite difficult. If you're designing a single antibody to be able to neutralize all different possible mutations, that's impossible. But are there ways that you can design a cocktail, very few of them, 2, maybe 3, that acts by different blocking or neutralizing mechanism, and I think that will have a better chance. And this is something that we are also considering as well. So for example, our 5A6, fortunately, it has pretty broad neutralization spectrum, but it missed the strain from South African, and it works pretty well for Delta, works pretty well for a British strain, but for the South African strain, it is much less effective, and this is something that we have to be concerned about. We have to be aware that that this can happen, and then how do we use our antibody engineering technique to engineer something that had a broader protection spectrum, and that's something that is our focus now.
And I just I want to remind everybody that if you have any more questions, feel free to put them in the comments box. And wondering, did any of you have Dr. Chang or Dr. Craig anything else to say about vaccines, the implications of this for vaccines?
This is probably more in Charlie's space than in mine.
Yeah, Robin, we think about it a lot. So, you know, I think Ching-Yi really covered the key points here, and that is that for vaccine design, first we have to find where are the right spots to develop antibodies to, right? The technology for putting, you know, RNA into an individual, transfecting them with RNA with the spike protein and generating antibodies to that is phenomenal that's happened. Now, how do you engineer the right substitutions in that? Because just putting in wild type isn't going to help with all these other variants that are coming up. So we clearly have to pay attention to these RBDs that are going up and down, right? That's absolutely important. So getting them to be locked down, up, that's going to be one thing. But the other, which not only us, but the entire field is working on, is that the spike protein is, you remember that white model that Chang-Yi showed, the RBDs were just at the tips of it. But the rest of that spike protein, that it's a gigantic trimeric protein. Right. It's enormous. It's not easy to make, right? So it's dynamic. So those tips up there are interacting with the arm down here. And how some of those mutations aren't up here just in the tips, some of them all the way down here in the arm. So how does that mutation down here affect this that's going on up there? I wish I had the answer, but it makes it extremely exciting to continue to try and probe how do we get at these dynamics, and the structures are going to be key to that. But in the end, you got to find something that's going to work by making the right antibodies. So it's like working with gloves on. You're not directly making it and putting it in. You got to make the immune system come up. And then you also have to think about the T cells, because you have two arms of your immune system, the B cells and the T cells. And I know Ifan recently had his antibodies checked, and he has a stronger T cell response than he does a B cell response, right? So now we got to think about that. So we're all learning immunology.
And biology is so complicated. So we just one final question for you guys. Where is kind of your next directions with this? What are you looking at now? What are your aims for the near future?
Go ahead, Chingy, you want to start?
Yeah, for the future, I think that this is something that Charlie's students, and I guess Ivan's students as well, will be working together with us. So we're considering a different way of antibody generation, and Charlie brought up this a more advanced way, but by data-based, and this is probably something related to what is that called? Google Fold, right? And so we
AlphaFold.
AlphaFold, I'm sorry. And so we're trying to see whether a collection of antibodies structures can give us a hint how we can design an antibody, not by random selection, but rather with a rationale to it. And this would I think this has potential to drastically improve our antibody potency based on this is knowledge-based, structure-based. And I think this is a great tool, because we can think about how, just like Charlie said, with a certain conformation, certain structure of the spike that we want to target, and based on this structure, how do we rationally design something that would fit into that the spatial conformation? And this is a direction that we're heading to.
Ifan, you want to go next?
Yes, so I feel for me this work was actually very exciting, and then in multiple different levels. One is, of course, we feel like we made a contribution and to the fight against the pandemic. Also in the science side, that we also, particularly for me, I used to think about antibody binding, basically think about think about where it binds and what's the direct effects of antibody binding. But this one also made me start thinking about really the antibody, how does it change the 3D conformational language of an antigen, of the target protein. And then that's also another thing that to consider beyond we just look at the antibody binding. So like Charlie, like Cheng-Yi, three of us was keep thinking about what is the better way to generate an antibody. And then not only just one thing to think about how these antibodies bind and how to improve the affinity, but also the other level is to think about how does antibody really to the conformation of the or trapping the conformation, alter basically landscape of the conformation of the antigen. And then this is really beyond simply beyond the SARS-CoV-2 spike protein, but probably the similar thing can be used for many different diseases when the antibody can be developed as a drug. Then the antibody basically is like a really beyond the small, is a major category for development, therapeutic purpose. And we used to use antibody as a tool, particularly for me, as a structure, we used to think antibody as a tool, but now we also think about the to emphasize or putting the efforts into how can we have a better way and then to push the antibody development itself. And particularly in the new era of biology, basically is this AlphaFold, which can predict the protein conformation pretty. These are very exciting things that happened just in very recently, but also similar sort of machine learning based, sort of deep learning based artificial intelligence. This is a way to think about the design and then to improve the antibody. This is also something that we've been keep thinking about it. And yeah, but this is kind of like a general direction that which collaboration lead us together thinking about a question that which is actually beyond the current level of this specific target, but a more broader term. Yeah.
Okay, I'll try and be quick. It's a big question, Robin, and I've always enjoyed trying to develop tools to help other people answer important questions. I draw the analogy to making a hammer that drives the right nail. And so part of that is, in this case, is always finding the right molecules. And finding these really special molecules that then can do things and can enable some of this structural information is something that when Chang-Yi was in the lab as a postdoc, we had that enjoyment to do that back then in early days of protein engineering. And now one of the things that fascinates me is that the library that these antibodies came from, and Chang-Yi built one in my lab when he was in my lab, and then the one that generated the 5A6 and the 3D11, that's a 10 billion member library. Now, 10 billion is a big number to comprehend, right? And finding 5A6 out of that 10 billion, it really is, it's not 1 in a million, it's 1 in 10 billion, all right? Finding that is really fascinating to me. Finding that special one that can do special things like this is something that I'm absolutely fascinated by. And Dong-Hee in the lab now, actually, Yifan called me last night and said, hey, can we get together and talk about this new target? So I met with him early this morning to talk about how do we find that really special molecule to go after this really difficult target that he has now. And I'm sure we're going to bring Chang-Yi on this as well. so that's, anyway, that's a little snippet of it. And I wanted to thank you for being such a wonderful facilitator and host for us. And thanks to all of QBI.
Sure. Well, that's a great place to end this great conversation. We're at the end of our time, but this has been really interesting, and thanks to our guests for all and thanks to Dr. Chang and Craig and Wang for being so interesting. And if you want more QBI TV, we do this with some regularity, so subscribe to us on our YouTube channel, and you'll find out when we're doing new things. So thanks, everyone.
Thank you.
Thank you. Take care.
Right.
All right. See you later.